The goal of this procedure is to describe a push-pull method for collecting blueberry volatiles and to investigate the response of undamaged blueberry branches to volatiles emitted from herbivore-damaged branches within plants. This is accomplished by first collecting local and systemic volatiles from blueberry plants damaged by gypsy moth caterpillars, mechanical damage, and by an exogenous application with the phytohormone methyl jasmonate. The second step is to expose undamaged blueberry branches to volatiles from branches damaged by the herbivore gypsy moth, and to then collect volatiles from these exposed branches before and after herbivore feeding. The third step of the procedure is to extract the collected volatiles. The final step is to inject the extracted volatiles onto a gas chromatograph. Ultimately, results can be obtained that show induction of blueberry volatiles by herbivore feeding and their role in intra-plant signaling through a push-pull volatile collection system and gas chromatography.
The main advantage of the push-pull system over other existing techniques such as the solid phase microextraction or SPME is that the collected volatile samples are desorbed at volumes that allow for multiple analyses. This method can help answer questions in the field of insect-plant interactions, such as the role that plant volatiles play in plant defenses and also on pollination.
To induce volatiles by herbivore damage, first bag 2 branches of blueberry plants with a spun polyester sleeve. Then place 3 gypsy moth caterpillars in each bag and allow